Thank you, Jeff, so much for the introduction, and thank you for the invitation to speak to the CAB today on the role of the microbiome in HIV cure research. So first, I'd like to take a moment to especially thank everyone for joining today on a holiday in the US, and also take a moment to appreciate Martin Luther King Jr. I found this and thought it was very apt for the HIV research community and the HIV community at large, that whatever affects one directly affects all indirectly. And I think that this is very, very true in the context of HIV cure research. So just a quick summary for the community, and it's okay if you don't understand this right now. Hopefully by the end of the talk, you'll understand what all of this means. So the key question we're asking here is, what is the role of the microbiome in establishment and persistence of the HIV reservoir? Our key finding, and this is going to be mostly preliminary data as I'm explaining what we're doing going forward with the RID HIV Mark Delaney grant, but we are hopefully going to convince you that dysbiotic microbes and or their metabolites in HIV may directly or indirectly contribute to viral persistence, and this is likely through inflammatory processes. And why is this important for HIV cure? Well, elucidating the role of the microbiome and the metabolites and the pathways involved may provide novel targets to improve cure strategies, which is ultimately our goal.
So just to make sure we're all on the same page, I'm going to give a little background on the microbiome. So the microbiome is the microorganisms in an environment. There are 10 to 100 trillion microbes in a person. We tend to think of things like bacteria as gross, but in fact, they are extremely important for maintaining health. The microbes can include things like bacteria, which we talk about the most, but it also includes viruses, fungi, protists, and archaea, and also the genes, metabolites, and products that they make. So these bugs are not just sitting there doing nothing. They are very actively metabolizing things like fibers, all the food products we eat, even the drugs we take, which we'll talk about briefly. So they're extremely important to human health. And they actually outnumber us. So there's actually more bacteria cells in our body than there are human cells, which seems a little crazy, except for when you think about the fact that a bacteria cell is much smaller than a human cell. But I always love to say the fun fact, and anyone who's heard me talk has heard this by now, but the average adult has 5 to 10 pounds of bacteria in your gut. So you can talk about that over your next dinner. Gross everyone out, but it's really important, and we want that. An important word that I'll use a lot here is dysbiosis. Dysbiosis is when you have an imbalance in this microbial community, and this can be an imbalance in either the abundance of different bugs or functional changes to these bugs. And I tend to call them all bugs, whether it's bacteria, virus, whatever. But this talk today, we're going to focus on bacteria. There's a lot of work being done, especially on the virus and fungi side and HIV. These fields are a little bit behind the bacteria side. We're going to focus on bacteria today, but I just want to point out that they are very important to study the other microbes as well. So pretty much everything in life can impact our microbiome, as we continue to see. And then there are multiple microbiomes, which I'll talk about in a moment with dysbiosis too. But so, and really this slide, so this is from a review, but I think that this should actually be kind of more circular, because the microbiome can also come back and impact all these things as well. But things from genetics to environment, to diet, lifestyle, hormones, industry, they all can affect the microbiome. And then, sorry, I'm moving these pictures, keeping it away, so I'm just moving that over. And then we have, all these microbiomes, and it's extremely important for these microbiomes to exist and do things like lubricate our pulmonary tissues, which we need that. They, help ward off pathogens, they assist in digestion. It's extremely important for things like complex carbohydrates in the colon that wouldn't actually be digested if it wasn't for our microbes. In the vagina, for example, microbes can maintain our pH and make sure that we don't get STIs. So there's multiple roles of microbes. And when we have dysbiosis, as I talked about, and these go wrong, and this is just a short list of the many, many, many diseases that microbiome dysbiosis can affect, but there's a lot of consequences when the microbes go wrong. So the gut microbiome is definitely the best studied, and it's the largest microbiome in the body. Definitely the vast majority of bacteria cells are in the gut. However, as I showed, there's multiple organs they're at. and they can result in massive dysfunction and health complications when we have dysbiosis. And we're going to talk about that a lot just in the context of HIV today. Another thing, and you're probably all familiar with the concept of inflammation, but you know, the microbes go hand in hand, and we'll go back to kind of how this is kind of a circular pathway. But inflammation, I always say, is the crux of all evil. It really is the root of most disease, including HIV, where we know that chronic inflammation is one of the key factors that leads to morbidities and mortality in HIV infection. And so it's extremely important to understand this in the context of the microbiome. And then, of course, immunity, which I think we know better in the HIV field than most fields. You know, the whole world has gotten a lesson on immunity, or half the world has not gotten that lesson, but many of us have in the last, couple of years. We've heard more about immunology than before, but I think the HIV field has been specifically, well primed on HIV, and especially in the context of what happens when we lose CD4 T cells. But these are all very intertwined and extremely important for overall health. I just want to take a quick minute to talk about methods to modify the microbiome, because this is one of those challenges with the microbiome. It's super easy to modify. So we know antibiotics, they're definitely miracles and kill lots of things, but we also have antibiotic resistance and overuse of antibiotics. So it's not a simple issue, but antibiotics, they can kill and limit bacteria, but they aren't very specific, and they have a lot of unintended consequences. And if this is an interest of you, we've done a lot of work here. I'm not going to talk too much about it, but I can certainly talk offline if you're interested in this. We have probiotics. So probiotics are live bacteria, yeast, microorganisms that are beneficial to health, and they typically reduce inflammation and promote health. I've done some work in SIV-infected monkeys showing that in SIV-infected macaques, it might decrease inflammation and increase CD4s. However, there was an ACTG trial with probiotics in HIV-infected individuals, and there was not a whole lot seen there, unfortunately. There were some mild differences in CD4s and maybe some slight reductions in inflammation. But the probiotics can be problematic because they don't graft well, and typically you have to just give so many that it rarely makes it through the stomach and alive through the system. So this is still a field that we're learning a lot about. Prebiotics are then food to promote the growth of beneficial bacteria. These are typically dietary fibers. And then you might have heard of fecal material transplants. So this is actually when you transplant feces or fecal material from a healthy donor into someone with a compromised microbiome. This is mostly used in C. diff infection. There's some moderate effects shown in HIV, but overall, it hasn't been, the magic bullet that we want it to be. So I think this is a good overview of the microbiome being its own ecosystem, needs care as such. So, it shows, you have, your own ecosystem, but if you give antibiotics or you have an infection or something happens to alter that, it's devastated. And if you don't kind of prime it, it can be left alone, and you get these weeds and you don't get this nice lawn that you're looking for, right? So the kind of what I just said, you have a sort of probiotic, so you seed the good microbes. You can put prebiotics on first to help those seed better, or this bacteria therapy, so like a fecal transplant. And you hope that then you can have this, beautiful lawn again. Like I said, it's not that simple. There's some things in the work that, we're trying to understand and trying to improve these therapies, like targeted probiotic therapy, live biotherapeutics, which have some promise, especially in something like lactin V for the vagina. There's some engineered probiotics underway, and then a lot of things targeted. Phage therapy is really big, especially out there in California. Steph Stradley at UCSD is doing some really amazing work with phage therapy. But overall, I think there's a big problem that we just don't understand the microbiome well enough. And this is where I think it becomes really important for the research we're doing, because I think having very specific targets is really what we need. And so that's really kind of the goal of trying to better understand the role of the microbiome in HIV pathogenesis and cure research, so that, potentially we can find, very specific targets when we know that the current strategies to alter the microbiome are not great. So I like to show a slide. This is kind of an overview of how my lab looks at things, and I think a lot of teams and I should mention too, Rafik Sekley is actually the lead on RF1 now, and then this, we both look at this very similarly and work together very closely on these things. But it's really this kind of vicious circle where you have inflammation and mucosal dysfunction, microbial dysbiosis, and immunity and inflammatory responses, and they are all intertwined. And so we're really trying to tease out and understand how they're intertwined and what are the key factors that maybe we can intervene in to improve health. So just to back up, and I'm going to go through some preliminary data for why we know this is important. So we've long known now, and there's been a lot of people who've done a lot of amazing work demonstrating this. But basically, and this is an old slide number on the bottom. I do not have 77 slides, by the way. That just is a slide number that's snuck in from another presentation. But we know that inflammation, and particular inflammation that's associated with gut dysfunction, is a key predictor of morbidities and mortality, like I said earlier. And all of these inflammatory processes tend to go back to the gut in some way. So whether it's microbial translocation or things like zonulin or IFAB, or IFAB, or looking at different markers of gut dysfunction. This is just a little cartoon though that kind of demonstrates what's happening. So on the left side, you have a healthy gut, and you want that intact epithelial barrier, and that keeps pathogens from crossing. You have homeostatic cells and cytokines like nice CD4s, now 17s, that promote homeostasis and promote that barrier and barrier health and mucus production, and have a nice crosstalk with the microbiome, making sure everything stays healthy. We know that these Th17s, among other CD4s, are depleted in HIV though. And then we move to the right-hand side here. We have pro-inflammatory cells and cytokines. I'll talk briefly about things like neutrophils that are highly pro-inflammatory and can break up mucosal barriers. You end up with damage to that tight epithelial barrier that can allow microbial translocation. And then you have this altered microbiome that we'll talk much more about, that the microbiome then can translocate. And then you have this microbial translocation that goes systemic and can cause undue inflammation and morbidities and mortalities. So there's been a lot of work. This is just kind of a summary from 2019 of some of the, trying to compound all the changes that we've observed in HIV infection and the changes to the microbiome that occur. We often talk about it, the microbiome is dysbiotic. So if it's a green arrow, it's down. If it's a red arrow, it's up. We're not going to go into the specifics of what bugs. Happy to talk more about this if anybody has questions, but we'll kind of talk about it as we go through, and I'll talk more on the next slide. The other thing I'll talk about this again in the next slide, but MSM has been kind of a controversy with this whole field, where it's been shown that receptive anal intercourse itself can alter the microbiome. However, it is very clear that that is not the only cause of altered microbiome. So people who do not engage in receptive anal intercourse and are HIV infected still have dysbiosis. So while that might contribute to HIV transmission, it does not seem to, or it is not the only factor that results in dysbiosis in HIV infected. So just the, kind of an easier summary of this is an increase in HIV. What is consistently observed is increased Prevotella. This is important because Prevotella are very inflammatory bugs. They have a very long LPS chain, and they tend to stimulate, especially innate immune cells. So I'll show you momentarily. We actually have evidence that shows that Prevotella can directly stimulate neutrophils, for example, and cause neutrophil accumulation. And then the neutrophils are very inflammatory and might be contributing to breakdown of the GI tract. We also see increased proteobacteria. This is also important because proteobacteria contain opportunistic pathogens. So these include things like staph and E. coli. And proteobacteria also tend to be more adherent to the mucosal barrier. And so we also find proteobacteria translocating across that epithelial barrier into systemic circulation and can be found in different organs. So the fact that these two bugs are consistently found to be increased, these two are consistently found to be increased, is really important for HIV. And then we find some of the good bugs that we want to see are decreased in HIV. So we have a loss of firmicutes. So this includes things like our probiotics, like lactobacillus. And then loss of bugs like Bacteroides, and this is important because Bacteroides are one of the biggest makers of something called short chain fatty acids. You might have heard of butyrate, one of the short chain fatty acids that's been probably most well studied, but these are important byproducts that are made metabolites of the microbiome that are produced by microbes and extremely important for maintaining health. And like I touched on, there was this paper saying that the microbiome is linked to sexual preference in HIV infection. While there's certainly some associations there, it is not the only factor leading to dysbiosis, for sure. I think we've cleared that up effectively at this point. So now, as promised, I'm going to go through some of the data that has been shown that, leading up to why we think it's so important to study the microbiome. I'm not going to go into detail on the figures. Don't worry about it. If anyone has further questions, or if anyone wants me to walk them through later, or wants the papers associated with these, I'm happy to provide more details. But I'm more trying to just make the points, but give you some, pretty pictures to look at from summarizing lots of hard work by lots of scientists. So the first one comes from Danny Duick's lab, and this is actually a collaboration from multiple people also involved in Red HIV, including myself and Rafiq. But they just recently published some cells showing that actually they can measure microbiome in the plasma by doing very deep sequencing, and the constitution of the microbiome that's in the plasma, so in circulating an HIV infection, is associated with immune reconstitution after ART treatment. So this is really good evidence that these microbes are having an effect, they're getting across in HIV, and that there's an effect on immune reconstitution. I mentioned we've done a lot of work with neutrophils. On the left here, these are just plots showing HIV negative versus HIV positive microbiomes, where we do see a difference. And this is actually after controlling for age and sex and sexual preference and gender. And we still see this association despite sexual preference. But what's really important is that there's a significant negative correlation between these bugs. So I mentioned Lactobacillus being a good bug, and then Prevotella being bad bugs. And when we look at this Lactobacillus to Prevotella ratio, there's actually a significant negative correlation with the ability for neutrophils to survive in the gut. These are actually gut colon neutrophils that we pulled out from HIV infected people. And the neutrophils can survive longer when this ratio is lower. So when there's more bad bugs, the neutrophils survive, which then can break down that barrier. This is work from Rafiq Sekali's lab, and he's done a lot of really nice work showing that you can, when they look at different species of lactobacilli and then the metabolites that are made from the microbiome, such as these butyrates and these different bile acids, that it's actually associated with inducible HIV reservoir. And that you actually, you have the hydroxybutyrates and bile acid metabolites. These are, again, metabolites directly made by the microbiome. The only thing in your body that can make these is actually the butyrates in the microbiome. The bile acid metabolites are associated with liver function as well, but they're positively associated with inducible HIV reservoir. They can also induce things such as these monocytes, and that's also associated with this inducible HIV reservoir. So again, showing this, association and link between the microbiome and the reservoir. And then we also, these are data from Ramit Sharma and Rafiq's lab, and so it's kind of Rafiq's lab, and we also have data from my lab. But like I said, antibiotics can have very negative consequences, and they showed here that. And we showed very similar findings as well in monkeys, but I guess these are monkeys as well. But it's, what's very clear is you get these increased innate cells, especially inflammatory innate cells such as neutrophils, and then decreased Tregs. Tregs again are anti-inflammatory T cells that you don't necessarily want to lose when you have an inflammatory condition like HIV. And antibiotic use can, and altering that microbiome can cause these disruptions. One more thing that is we're not necessarily looking at Red HIV, but something that I'm working on separately and closely with Rafiq, and then we're working together with the Red HIV, is the concept of that microbes can actually directly metabolize ARVs. So we showed this using vaginal bacteria, that this might affect PrEP levels in women. And now we've moved on to we're really focused on the gut now and how dysbiotic bugs, I showed you how dysbiotic the microbiome is. Well, we know those microbes specifically that are dysbiotic are also capable of metabolizing antiretroviral drugs. So we're working very closely to study this and working with the MDC teams to understand if direct metabolism by bacteria of the antiretrovirals might be associated with increased inflammation and altered HIV reservoirs as well. So I hopefully I've convinced you that there's several factors that we've identified in my lab, Rafiq's, and many others that really show that the microbiome might impact HIV reservoirs, and the importance of studying this and understanding kind of the role of the microbiome in HIV persistence. So that's kind of the goal of RF1, and direct and metabolic induced effects of microbiome regulation of innate immune dysfunction and HIV persistence is the title. Like I said, Rafiq Sekhli is the lead. Daria Hazuda at Merck and I are co-leads. Gatul Sonmanithan was working closely with us, but has now since left Merck. But so we have a large team all working together on this. So the aims of this project, and these have altered a little bit just based on funding and things, but it's overall these are being done one way or another, whether through RID HIV or peripherally. But we're all one big team with the same goal. So I'm just going to keep them as we envisioned them originally. So so the first aim is the role of microbiome-mediated metabolites and modulating innate immunity reservoirs. So really diving into what is the function of these bugs and how does it modulate the immune system and potentially have an impact on reservoirs. Aim 2 will look at the microbe directly inducing pro-inflammatory innate immune responses and how that might affect reservoirs downstream. And then in aim 3, we are using aim 1 and aim 2 to identify host receptors and pathways that are critical that the microbiome might influence for latency reactivation and establishment. I'm just going to take a minute to go through each of these. Oh, I'm sorry, I'll go through those in a moment. But so we just wanted to point out we have for part of RID HIV and MDC at whole, and then other studies too that are just contributing to this that aren't necessarily funded by MDC, but like I said, it's all one big team with one big goal. So we have a lot of HIV and ARDS-treated patient samples. And then we also have NHP models, both retrospective and prospective cohorts for both of these that we're able to investigate these questions with. So in aim 1, our goal will be to investigate the role of the microbiome-mediated metabolites. So these are things like the short-chain fatty acids like butyrates and then bile acids, and how they may modulate innate immune function and HIV latency. We hypothesize here that these metabolites contribute to dysregulation of innate immune cells, and that impacts the HIV-1 latency and or reactivation status. And the deliverable for this is hopefully to have in vitro identification of key metabolites and genes that are important for HIV reservoir seeding, establishment, and persistence downstream of bile acid and butyrate signaling cascades. I know that's a mouthful. Don't expect you to remember all of it, just trying to get kind of the overall points across, and then we can discuss more in detail. So for the aim 2, we're breaking this one now down to look at the direct effect of the microbes. So in aim 1, it's really about what are the microbes doing, what are they making, and what of those metabolites that they actually make and the function of those microbes, how does that affect latency in reservoirs. In aim 2, we're looking at the microbes directly, and we have many tools to be able to do this and how they modulate innate immune function, and then have and HIV latency. And this is where these LARA assays are important. We do a lot of co-cultures. With Merck, we have some really amazing tools with bioreactors that we can study these now. The tools have really improved over the years. Here we hypothesize that dysbiotic microbes in HIV infection lead to increased innate immune cell inflammation and accumulation, and this can drive increased HIV reservoirs. And so the deliverable here again is identification of microbial species that can directly modulate immune function in HIV reservoirs. So again, kind of the goal of RF1 is to identify metabolites that are important and what pathways they trigger. And the goal of aim 2 is to develop or to identify direct microbes and the pathways that they can trigger that can associate with the persistence and latency. And ultimately, those two aims will come together where we hopefully identify these different microbes and metabolites that are really important in HIV. And then in aim 3, we're going to mechanistically validate these. So now we are going to use a combination of NHP models and co-culture and bioreactor systems, where we can take the different bugs that we identify or the metabolites from those bugs that we identify and actually go in and understand how they influence latency establishment and reactivation. And we hypothesize that if we alter these and target the metabolome and the microbiome, we might be able to modify SIV reservoir levels using an HP model. And here we have the deliverable both in vitro and in vivo validation of antibiotic and microbial transplant methods, small molecule compounds, antibodies, and or other pharmaceutical tools for novel therapeutics. So, and this is where it becomes really important that we're working so closely with Merck. And we're really fortunate to have someone as amazing as Dr. Kazuda working with us, because they, they can really take what we're identifying as academic researchers and really put this into a drug development system. So hopefully we can find something. And here's just an example of, how we envision these monkey studies to go, where we have, these different scenarios where we can try different agonists and antagonists that Merck would make, or we can, decide which beneficial bacteria we've identified that have the best effect and use those specifically in a microbial transplant. And try these different things to see if we are altering the microbiome and using very targeted approaches to alter the microbiome in SIV-infected monkeys on ART, can we have an impact on those reservoir levels? So that's really going to be the ultimate proof of concept. So this is just kind of a nice overview that I think, hopefully will kind of wrap up a lot of words I just threw at you and what we just said. But so for this study, the overarching view of it is that we have human and NHPs and co-culture systems. And, the ultimate kind of deliverable is, identifying things that impact this latency and reservoir of HIV. And we believe that these are all these very intertwined pathways. So you have the microbiome and the different microbes that are dysphatic. We have that metabolome from the microbes, like the short-chain fatty acids and bile acids, and that these can independently or both together lead to innate cell activation and inflammatory milieu that then all can impact, adaptive immunity and CD4s and these latency and reservoir. So just going back again to my original slide, hopefully I've convinced you the importance of the understanding the crosstalk and how the inflammation, mucosal dysfunction, microbial dysbiosis, immunity, inflammatory, all can be intertwined and really important to kind of tease out the mechanisms underlying this for hopefully having better therapeutic approaches. So back to my summary, hopefully this makes more sense to you now if it didn't in the beginning. Our key question is, what is the role of the microbiome in establishment and persistence of the reservoir? Our key finding here from our preliminary data is that dysbiotic microbes and or their metabolites in HIV may directly or indirectly contribute to biopersistence, likely through inflammatory processes. And how is this important for an HIV cure? Elucidating the role of the microbiome and the metabolites and pathways involved might provide novel targets to improve cure strategies. And again, our really great partnerships here, I think, will hopefully lead us to having deliverables that are potentially, targeted approaches to alter this. So I want to thank everybody on the Red HIV team. Smeet Chanda, Paula Cannon, Rafiq Sekli are our leaders. Rafiq is really head of this RF1 project, but it's we work very, very closely together. So like I said, Daria from Merck is really critical in these therapeutics and a lot of the really amazing co-culture bioreactor systems. Ashish Sharma has been working very hard on a lot of the preliminary data and is very integrated into RF1. Mirko Paradini is running all the monkey studies that I showed you, but the entire team is really quite amazing. And then again, thanks to Jeff Taylor, who I am just thrilled we have running our CAB, and I am really excited to work closer with the community on this. A quick thanks to my lab and their funding sources for the preliminary data, but I will leave it here and be happy to take any questions.
Terrific. Thank you so much, Nikki. That was an amazing overview. And again, amazing that you were able to do that despite battling COVID at the same time, battling two pandemics at once, right inside of yourself. So kudos to you. So now we'll open the floor to questions for anyone. I see Linda has your hand up. So go ahead, Linda.
Okay, wow, that was amazing. I can't believe how good you look. You sound a little hoarse, but you look great. Thank you for coming in and on the other, how do they say, over and above the call of duty. So it was really wonderful to
Jeff
see.
was motivated. We'll give Jeff the credit.
I know him well. I know how much he can be. It was really wonderful to see this all in one place, because I've often wondered, is it the number of immune pathways in the gut, but it's that and it's all this dysfunction that you're talking about and that you described. And let me just ask you something preliminary. Do you think that it's because of all these immune pathways? I mean, once HIV gets to your gut, in 20 minutes, it's everywhere else, Can you maybe touch on that? And I was also wondering what interventions you might visualize to deal with this dysfunction. Once you can prove that it's actually contributing to persistence, and what metabolites are and what bacteria or whatever it may be, what kind of interventions come to mind for you?
Yeah, so really great points and questions. So to touch on the first side, yeah, the dissemination of HIV is terrifying, really. So it's, I know Brandon was on here. It looks like he left, but because he's done a lot of really great work on that. But and it is, and I have long thought actually that there's a signaling pathway through the microbiome that we don't understand. This is something that I've been trying to work on for a very long time, that I think helps influence this. But it is, and what we do know is that very, very early in HIV infection, we see changes to the microbiome. So not only are we having dissemination of that HIV very early, but we're also having the inflammatory processes that can start changing the microbiome, changing that local inflammatory milieu, and causing that gut dysfunction happen very early. And we know that, from monkey studies, we know how early this happens from the acute trials in Thailand. Alex Schutz has shown some amazing data showing how quickly you lose Th17 cells, how quickly things change in the beginning. So it's a really important point, and I think something that we have to consider always and think about as reservoirs. As far as targeting, like I showed, the current ways to target the microbiome are just not greatly effective. And so this is where I think that this work becomes really important, because I think that it'll help us identify specific pathways. And, we have some ideas of these pathways that we can put into play now. If we don't find anything in Ames 1 and 2, we still know that butyrates are important, like the TMAO pathway. There's different pathways that are kind of our fail states that we're trying to put into place to target already. However, it's, I think, and, TGF-beta is a big one that we know is altered by the microbiome and by microbiome dysbiosis, and TGF-beta can kind of regulate other immune cells. So that's something that we're starting to target right away. But hopefully the goal is that we can identify very specific metabolites, and maybe we can come in and neutralize that. Or if we, identify something that's good, okay, well, we're seeing a positive correlation here that inflammation is going down and reservoir levels are going down if you make, fuconates or whatever it might be. And then maybe we can agonize that, and again, this is where our collaboration with Merck is so important. And can we, maybe induce a better outcome by, trying to, or add more bacteria that have that function, plus agonize it, trying different combination approaches. But that's where I think that we're going to make a difference. So it's, it's going to be a long path, but it's, we're working on it more and more so, and hopefully, soon we'll be able to alter things in a better way. But great questions. Thank you.
Thank you so much.
Great, thank you. I see Linos. Is that you with your hand up?
Yeah. Thank you, Nikki. Do you think there is a direct role of these EVs to be important in viral spreading, like not via an inflammation or on its own, or not via immune cells, but directly having viral particles or cellular cofactors into these EVs that play a role into spreading of the virus even under art?
Yeah, so that's a really good question, and it has not been shown at all in HIV. We've actually talked a lot about how to go at this, because you definitely can find virus mixed in with microbiome, but not necessarily directly attached, and our imaging tools just aren't quite good enough there. But so there is evidence, though. So in a mouse herpes virus, there's been some really cool data showing that it actually uses LPS made by the microbiome to infect cells and actually activate it. And for HIV, you need those cells to be activated to get infection and have productive infection. So there's, we've long had the question in my lab: is there a possibility that that the microbiome is directly acting on that? Is it using LPS, or is it using some other way of the microbe to get in and actually induce infection? None of this is proven. These are really tough experiments to do, but it's something we talk about all the time, and we're trying to tease out. And I actually just hosted a gut virologist to kind of team up with me and try to work on these questions, because I think that they're really important. But there's just no evidence either way right now. But I think it's entirely possible. So I think it's kind of probably outside the scope of these questions, but I think it's something that we can use the samples from these questions to try to get at. They're tough experiments, but I do think it is a potential. So it's a great question.
Yeah, I provide, so this in our university, there is Anne Hendricks. Maybe you can Google her name later on. She has been working on EVs a lot. Anne Hendricks. A-N.
Will you put the name in the chat?
Yeah, I will put it in the chat. And sometimes I provide samples where she's looking at things like that. She started looking in samples in cancer patients to look at how BEVs were present there, bacterial EVs, looking at HIV as a control. But what we saw was that HIV patients also had BEVs. So I will put it in the chat, because sometimes it might be interesting to team up for some of the experiments with her, because she has been working on it also. But I will drop it. Okay. And thanks for the answer.
That's perfect. That would be great, because I would love to get in touch with her. Thank you. Looks like Nelson put a question in the chat. Yeah. Besides fecal transplants, what other ways have been proven to effectively add viable prebiotics in the gut? Are there any formulations that survive the acid environment in the stomach? So there are. That's a great question. And they're encapsulating now probiotics and prebiotics to try to, particularly the probiotics, to survive the acid of the stomach and not dissolve till it gets in the gut. It's hard because a lot of these studies have shown that even despite that, a lot of the bugs don't survive and they're not engrafting. So there's a lot of work being done on that, onto better engraft with probiotics. But there are some things that and I take probiotics myself. Honestly, I always tempted. It's not going to hurt. There's very few cases that have ever shown, and they're like extraordinarily severe immunocompromised. And I think they're all children actually, but and they were like weird, really high dose probiotics. So I overall, again, I'm not an MD, it's not my medical advice, but I don't think because I think even if the bugs don't survive taking probiotics every day, a lot of those metabolites they're making, they still are in there, and still can cause anti-inflammatory effects. But overall, there are some other things you can do, like just even soluble fibers, being cognizant of your diet. It's been shown several times that dietary fibers and high fiber diets are associated with better microbiomes. And so just, being aware of that and trying to include fiber. And even, you can buy like the inulin on Amazon that can, promote better probiotics. And that's been shown, and that doesn't need to survive the gut. Now, other than that, there's different ways. So there's, I work with a company, for example, that's developing, probiotic enemas. So kind of, going from the bottom up in the vagina, suppositories. And then, like I said, there's a lot of, encapsulation techniques that are being looked at to try to beat those stomach acids, which are done with a lot of drugs, right? So it's kind of the concept behind a lot of the delayed release that, takes a long time to have those kind of peeled away, and it allows, 24-hour release for a drug. It's the same concept, trying to get those to the gut and have the probiotics released, where they should be, down the colon. So I see another hand raised.
Yeah, Uday, go ahead, unmute yourself and
Hi, Nikki. Great talk. I really liked it. And so I just want to apologize on behalf of my NIH colleagues there. I mean, today is an holiday. That's why you don't see so many people. So anyway,
Well,
I
I
enjoyed
appreciate that you made it, and all of you made
No,
it
I just
anyway.
love
And
this
it is
topic.
recorded, so we can share it.
Oh, wonderful. Yeah, I'm going to do that. Yeah. So it's an important topic. I used to work on it, at least some aspects of it. So when you talk about microbiome, right, I think people's main focus appear to be bacteria, but there's also viruses in them, the virome. So when you treat these patients, especially with antiretrovirals, some of them could have effect on the other viruses, which are commensal and has an effect on it. And when people talk about probiotics and these kind of things, it's always has a bacterial focus towards it. So I'm thinking, how important is the virome contribution, and what do you think about?
No, so, and that's a really great question, and I tried to touch on this in the beginning, but it is extremely important. So, and we just haven't had the tools that to study them well, but they have been done, and we are working on this very hard. So, Scott Hansley at WashU in collaboration, I think Doug Kwan was his big collaborator in that, showed a lot of, or showed that the virome is altered, especially in HIV infection, and that that virome might contribute to chronic inflammation, and that there's a different virome and there's expansion of certain viruses, and we know that that's really important. So we've talked about that. We actually almost included it in this study, but again, it started, then things become overambitious, and it could take an R01 itself just to look at that virome. So we are collecting samples, and we're doing some work in my lab, and then from the MDC, we'll also be saving samples and hoping to make a spin-off project to look at the virome and the fungome, which has been really underappreciated, which is kind of ironic considering that candida and fungal infections were one of the first really identified AIDS-causing illnesses. Yeah, it's kind of, we've regressed and not really gone back and studied that, when there's clearly a propensity for fungal infections that need this has susceptible to immune pressure here. So I 100% agree with you. It's extremely important. It's understudied, but we are on the pathway to better understand that, and then to understand those interactions as well. I think it's really extremely important.
Thank you.
Thank you. I see Linos's hand up. Is that from before, or did you have another question?
Yeah, sorry, I will drop my hand, sorry.
Okay, no worries. Actually, one question I had, you mentioned in your talk, doing some of the research in the animal models and the macaques. Are there any other animal models that might be useful? I know they use a lot of humanized mice in preliminary research, and has that been looked at? Or are they similar enough that could be useful?
Yeah, we've done some mouse work. I have a student that works between me and Mike Gale, who, and that's more on Zika infection and some other infections, and he's really identifying like vaginal microbiota that we can have a lot of overlapping. So if you're looking at certain times in the cycle, overall, it's been, humanized mice have been hard because the microbiome varies greatly and is very simplistic, and it's not the same. There's been some models. I know that they've been trying to kind of make a humanized mouse microbiome, and there's been some work on that. But overall, it's tough because what happens in mice, especially with the gut, tends to be that you put in like human feces, but they just outgrow one thing, and typically that one thing is not something that you'd see much of in a human. So that has been a challenge, but I think there's some good work on it. And then, if you guys aren't aware, this was actually worked on at University of Minnesota by Dave Massapus several years ago, showing so all these kind of, the mice that you get to use in the lab, from Jackson or whatever, they're very kind of like clean, not real life mice, whereas they went out and caught mice out in fields and found that those mice actually recapitulated human immunology and microbiome much better. So I know there's some work being done with those mice that, literally field caught mice. And so hopefully that will amount to something. But right now, I think that, humans obviously the best, followed by monkeys, followed by mice at this juncture. Of course, all of them come with caveats and expenses. So it's, I think for anything we do, it's always, taking the pros and cons and weighing what our best model is for the question we're asking. And so for microbiome, and that's why we've really, I used to do a lot more monkey work than I do now, because it's a lot of times if we can get the samples from humans, that's obviously going to be the best. But we obviously can't go in and do changes to humans that we don't know what is going to, happen because of it. So that's where, the next step is with monkeys, microbiome group comes in.
Mm-hmm. Great, thank you. Uday, did you have another question?
Yes, I have a follow-up question from what she said. So talking about samples from humans, right? So I think if you look at different parts of the world, the dietary habits are different. What they consume is different.
Oh, yeah.
So getting a sample across the whole world maybe, may offer a better understanding, I guess, right?
Which is a huge benefit actually of this MDC, and thank you to Linus and we and Vincent. But we have, for this MDC in particular, we have European and US sites, and then we also have other sites too. Like I said, that we all have kind of individual contacts and are doing things with that we'll be incorporating with the MDC as well. So like I always like to say, it's like we all have kind of our separate studies that might be funded for different mechanisms, but we're all, it's all in for the big win. So we'll be incorporating that. And, and I'm really fortunate that I have samples here that we've been working, we're getting samples locally in Minnesota. I still get a lot from Miami. I still get some from Washington. And then we have Thailand, Africa, Europe. So it's Mexico. So we have a lot of sites that, we're able to get samples from and understand this. And then on top of it, we try when we can to get, at least basic dietary information. So at the very least, like are you a vegan? Are you, keto? Things like that can have a profound impact. But you're absolutely right. There's a beautiful study actually done many years ago by Jeff Gordon's group at Washington University in St. Louis, where they actually looked at people in St. Louis versus people in Brazil. And that the people, I think it was Brazil, somewhere in South America, but that vegetarians in St. Louis looked more like a vegetarian in Brazil than a vegetarian in St. Louis looked like a meat eater in St. Louis. So, I mean, geography and diet both play a very profound impact. And it's something that we definitely, but again, this is, I think, one of the big strengths of our MDC and for Rid HIV is to have those multiple sites. So.
Agreed. Thank you.
Terrific. Anyone else have any questions or comments they'd like to make? We've got about 5 minutes left, so.
Send me back to bed.
Terrific. Well, thank you all for joining. And again, thank you so much, Nikki, for that wonderful talk and for powering through.
Oh, thank you all.
Thanks.
Jeff, you're amazing. Thanks, everybody. And please feel free
Thank you.
to
Get
reach
some
out
rest.
anytime if you have follow-up questions. Thanks.
And if we can ever help you,
Thank
let
you.
us know that too.
We'll run around and get samples for you.
Yeah, I'm like, be careful what you ask for.
Yeah, I get it.
Thanks,
All right.
Nikki.
Thank
Thanks
you all
again.
so much.
Thanks,
Have a
Jeff.
Thank
This
you
was
all.
wonderful.
Take care.